يلا سريع سريع عشان نخلص محاضرتين إن شاء الله. So, last thing we talked about. مش قاعد بالسينما. Definitely مش قاعد بالسينما. Okay. So, آخر شيء حكينا عنه هو إنه الهيموجلوبين can be glycosylated. And it's valine, interestingly, it's valine that gets glycosylated. This happens particularly when the level of glucose in blood is high. Okay? And that is why it has so far been used as a diagnostic marker for diabetes or to follow up the treatment of diabetic patients. Okay? So this is the major point. The thing is, there are two tests for indicating diabetes or pre-diabetes. The first one is total cholesterol level in fasting individuals. Okay? So they tell individuals, fast overnight and come early in the morning before eating anything. We'll do a blood test. And this is a test to see the level of glucose right at this moment. Okay? And it should be between 80 and 120. This is the normal range. Now, if it is a little above 120 to 140, they consider this as pre-diabetic. Now, for glycosylated hemoglobin, it is a test to indicate long-term glycosylation or long-term level of glucose, because it takes some time for hemoglobin to be glycosylated, and it stays for a while because now it exists in red blood cells. The half-life of red blood cells is about 120 days. So it stays for like 2-3 months. Okay, so it is a good indicator for long-term level of glucose. Whereas in testing in fasting individuals, this is like right at the moment. So there are advantages and disadvantages for both tests. Now there are currently what they do, there has been a change in numbers of or measurements or units for level of glycosylated hemoglobin. It used to be in percentages. That is, I think this is how much glycosylated hemoglobin, the ratio of glycosylated globulin to the whole population, the whole total hemoglobin molecules in a patient. And it should be, the normal level should be between 5 and 6. If it is a little above 6, 6 to 7, they consider this an indication of pre-diabetes. Above 7, this is like diabetes at different ranges. Now the new numbers that they consider is millimolar of glycosylated hemoglobin per mole of total hemoglobin. And it should be between 31 and 42, let's say. So these are the new numbers that are recommended. And I think that the new test, which is known as IFCC, gives a little more room or larger range than depending on, and it's more accurate than depending on small numbers like 5 and 6. So this is something that you will hit, inshallah, when you go to the clinic. So let's move on and talk about genetics of globin synthesis. So we said that we have two types or two classes of globin chains. We have the alpha and we have the beta. And we have the zeta and we have the epsilon, right? And we have the alpha and we have the gamma as well. So these are different populations of globin proteins. And it turns out that they exist as clusters. So on chromosome 16, we have the locus, we have the position or the gene for the zeta, followed by the gene for the alpha. And pay attention to this. There are 2 genes for alpha on every chromosome, and they are known as alpha 1 and alpha 2, and they are identical. Okay? And this is really important when we go to that lecture on hemoglobinopathies, because an individual in this case would have 4 genes, or let's say 4 alleles of alpha, because now there are 2 on every chromosome, so we have a total of 4. So that's one. On chromosome 11, we have the beta globin cluster. So it is a cluster that contains the epsilon, and we have 2 genes for gamma, and then we have delta, and we have beta. And there's only 1 beta. So every individual has 2 genes, or 2 alleles for beta, 1 on every chromosome. But for alpha, no, it is a total of 4. That makes a difference in diabetes. Now, it makes a difference in thalassemia diseases. Now, there is something else that is really interesting. The order of the genes reflects the order of expression. That is, we know that in the embryonic stage, we have expression of zeta, followed by expression of alpha. Now, for the beta globin, we have expression of epsilon, then expression of gamma, then expression of beta and delta, all right? And so the order is the same. We have zeta, then gamma, then we have, in the adult stage, we have both delta and beta. And it turns out, actually, when they studied this further, and when they studied the regulation of globin synthesis, and remember, it's really timed. So it turns out that even premature neonates, if they are born in their 7th month, they still have the HbF, the fetal hemoglobin. And the switch happens according to their gestational stage. That is, delta is expressed at the 9th month. Okay? So it's not like once they are born, you have a switch. No, they follow the same timing, okay, as a normal baby would be born. So that is important as well. So they looked into the genetics, how is expression of globin genes regulated. And it turns out that each one of them has its own promoter and regulatory sequences right before the expression start site or the transcription start site. So for example, for the epsilon, it has its own promoter and it has its own regulatory elements in their DNA. For gamma, same thing. Each one has its own promoter and regulatory elements. Same thing for delta and same thing for beta. Same thing for alpha and zeta. Now, for the beta globin, because it's maybe more well studied, there is an enhancer region that is located upstream. Remember terminology of molecular biology. It exists upstream of epsilon, and it's known as LCR, for locus control region. This is an enhancer that controls expression of all of these clusters. And it turns out as well that regulation follows different mechanisms. So we have a promoter for each one of these genes. Each one has its own promoter and regulatory elements, and they are all controlled by this enhancer, the LCR. Okay? So what happens is that in the fetal stage, you have these transcription factors, specific ones. Don't worry about the details. They bind to the promoter region of gamma, and they interact with this enhancer, regulating or inducing expression of gamma globin. Then later on, you have a switch where now we have transcription factors that sit on the promoter region of beta. Okay? And the enhancer region interacts, or the proteins that bind to the enhancer region can interact with these transcription factors, regulating expression of beta. So everything is timed, and it follows a certain program. Okay? And this is how control happens. So we have control by one enhancer. We have control by the chromatin looping. So the looping, notice that the looping is different in different stages. there is regulation by transcription factors. We have transcription factors for gamma, we have transcription factors for beta, we have transcription factors for alpha, and so on. And number 3 is epigenetics, which is basically modification of DNA or histones. You have methylation of the promoter region. Remember, molecular biology, methylation of the promoter region means that this gene is inactive. Okay? So when you have for the gamma globin gene, the promoter region is methylated, it means that it is silent. There is no expression. This is unmethylated, that is the epsilon, so there is expression right here. Now, when you have a transition from the embryonic stage to the fetal stage, now you have methylation of the promoter region of epsilon, so now it becomes silent, and this becomes unmethylated, it becomes active. So you have regulation at different levels, but eventually they are all controlled by this enhancer region. And this is really beautiful, because treatment, meaning what? If you listen to that lecture on hemoglobinopathies, if someone cannot synthesize beta globin or enough beta globin, well, why don't we rescue this person by inducing expression of the gamma? It functions well, there's no problem with that. So by understanding how expression of gamma globin is regulated, maybe we can treat patients with beta thalassemia, that is, patients who cannot synthesize enough beta. We can compensate that by making them express enough gamma globin proteins. Okay? So listen to the hemoglobinopathy lecture, and you can go back to this in order to understand it fully. Okay? هلا أختكم has read something about this, but this is outside the subject. Okay? Okay? Any questions, by the way?
Okay, so what I have read is basically an abstract of research titled The Control of Globin Gene Expression During Development and Erythroid Differentiation. So basically that in an eye-catchy phrase, إنه بحكي لك from various genetic and biochemical studies, it was realized that then the production of fetal hemoglobin is the reason why patients with severe beta chain deficiency remain alive. معناته conclusion, our take-home message, it became obvious that then abundant synthesis of fetal hemoglobin can cure beta thalassemia and sickle cell diseases. بالفعل كيف عن جد لو صار في switch where we can produce fetal hemoglobin instead of damaged adult hemoglobin, we can rescue such patients.
Good. Yes, I agree with you. Thank you. I'm happy لما أشوفكم هيك when I entice energy عندكم. أحكي لكم شغلة and you go and read more about it. It's fulfilling for me. It's satisfying. نعم. في اختلاف بين البيتا والدلتا. الدلتا is not expressed very significantly. والبيتا is the major one. من ناحية structure أكيد في differences, لكن major differences I have no idea. Is alpha 2 delta 2 functional? I have no idea. I haven't read anything about it. Okay, بس هذا سؤال. طب ليش delta is not expressed as much as beta? Because there's something wrong with its promoter. The promoter is not really that active for some reason. يمكن صار في mutation during الخلق تبعنا والتطور تبعنا خلال millions of years ago. ايش صار بالضبط؟ I have no idea honestly. بس that's the reason. بس إذا actually حلينا هذا الموضوع، إذا عملنا compensation إنه عملنا expression للدلتا والدلتا is functional just like beta, that's beautiful. بس نعرف كيف. Alright, so let's talk about regulation. So hemoglobin is an allostatic protein, and it is regulated by allostatic effectors. Okay, so these molecules, regulatory molecules, they bind to the protein. They change the structure of the protein, facilitating the transition from R to T or from T to R. So they can be inducers, they can be activators, or they can be inhibitors. And we said that oxygen is a homotropic allosteric effector. لأنه it affects binding of another oxygen molecule. So it's homotropic. Now, there are other factors that are heterotropic, so they affect binding of oxygen to hemoglobin molecule. Now, usually, these allosteric effectors bind somewhere else other than where the ligand binds to. Okay? Sometimes, like oxygen, they can bind to the same quote-unquote active site, or the binding site of the same ligand. So it depends on the molecule, but usually they bind somewhere else, like as shown in here. So you have a regulatory, let's say that this is the ligand binding site, the oxygen binding site. The regulatory molecule binds somewhere else. Right. So these are allosteric, that's allosteric regulation and allosteric effectors. So what are the allosteric effectors that we want to talk about in this lecture? Yesterday we talked about oxygen as an allosteric effector, and we talked about the different models of how oxygen would do that. So these are the ones that we'll talk about today. We have protons, hydrogen ions, we have carbon dioxide, we have 2,3-bisphosphoglycerate, chloride ions, and carbon monoxide. And we have something else that I didn't write here, which is temperature. So let's start with the first one, protons. It turns out that the affinity of hemoglobin towards oxygen is influenced by pH. And it turns out that normally at pH of 7.4, which is right here, the green line right here. Now, if you go to the right, affinity is reduced. If you go to the left, affinity increases. ليش? Because it has to do with the P50 value. P50 is what? The pressure of oxygen when 50% of the oxygen binding sites are filled with oxygen. So if you have more, if you need more oxygen to fill 50%, this means that affinity is low. If you need less oxygen, it means that the affinity is high. Okay, remember that. So normally, this is how it looks like, sigmoidal as well, right? It turns out that if you reduce the pH, 7.2, 7, 6.8, the curve shifts to the right, meaning that the affinity is reduced. Okay? So, and it does make sense as well. I know we want logically, think about it logically, not biochemically. It makes sense because in tissues, pH is lower than in lungs, right? Because in tissues, we have a lot of metabolism. And as a byproduct of metabolism, we have protons. The environment is acidic. So we want hemoglobin to have lower affinity for oxygen when pH goes down in tissues so that it releases oxygen to tissues. So this is an enlargement of the same thing. Right. So what is the mechanism of action? How do protons do that? And by the way, this is known, the effect of pH on oxygen, on affinity of oxygen to, affinity of hemoglobin towards oxygen is known as the Bohr effect, because there's a scientist known as Bohr, B-O-H-R, Bohr, B-O-H-R, okay? He is the person who found out that pH affects the P50, okay? And the question is, so what is the mechanism of action? It all has to do with the electrostatic interactions. So what happens is that when pH is low, you have more protons. These protons will bind to histidine. It's not the distal and proximal that we talked about yesterday. No, it's a different histidine. It is the last histidine in the beta chain. It's numbered 146, so it's known as histidine 146. Okay, so you have this histidine, you have the beta chain, the very last histidine, and the very last residue is a histidine, histidine 146. It gets protonated because now we have a lot of protons. And when it gets protonated right here, it can make an interaction with an aspartate. There's a positive charge, so it can interact, it can form electrostatic interactions with a negatively charged amino acid. Okay? Enforcing, strengthening the electrostatic interactions within the same peptide. Since it is also the last amino acid, it has a carboxyl end right here. And this carboxyl group can also form electrostatic interactions with the alpha chain, with a lysine on the alpha chain. So now we have electrostatic interactions, two electrostatic interactions that take place. One between histidine, the last histidine of the beta chain, with aspartate on the same chain, and with lysine on the alpha chain, the opposite chain. Strengthening the electrostatic interactions, and that results in facilitating, making it easier for hemoglobin to switch from the R state to the T state. Remember, T state, you have electrostatic interactions. When you have this transition from R to T, now it's the T state, low affinity, oxygen is released. Okay, so that's the mechanism of action. So that's one. That's the Bohr effect, really important. Now, the question is, where do protons come from? Well, they are byproducts of metabolism, that's one. Two, they also come from carbon dioxide. Remember, we have Krebs cycle, right? We have production of a lot of CO2. And this CO2 can combine with water, carbonic anhydrase, converting it to carbonic acid. Carbonic acid immediately dissociates into the ions, protons. So we have protons that are produced. So the main source of protons is actually CO2 as a result of that. And this is true. So if you look at this plot again, right here in red, we have pH 7.6, 25 torr of CO2. When you increase the level of CO2 from 25 to 40, there's a shift of the plot to the right, meaning lower affinity. Okay? Now, add a little more CO2, 60 torr. pH goes down to 7.2. Okay? So it shifts again to the right, lower affinity, and so on. So this is the effect of CO2. So someone might say, لحظة شوية. احنا هون we have two variables, right? And the first variable is the pH. The second variable is the CO2. So is it that CO2, does CO2 affect hemoglobin directly or indirectly? Indirectly meaning that that CO2 increases the concentration of protons in tissues. And so they studied it. Further, and they found out. They found out that actually it's both. It's both. There are two factors. One, it does so indirectly by increasing CO2. You also affect the pH. That's one. And it does so indirectly by producing protons. I'm sorry, let me say that again. It does so by two mechanisms. The first mechanism is that you increase CO2, you increase the concentration of protons, and that does Bohr effect that we talked about. That's mechanism number one. Mechanism number two is that it does affect affinity directly by interacting with the hemoglobin molecule. So it turns out that hemoglobin, the function of the primary function of hemoglobin is one, transporting oxygen. The other mechanism, or the other function of hemoglobin is to transport CO2 from tissues to lungs so that it can be exhaled. And how does it do that? By binding it directly with CO2. So what happens is that CO2 in tissues, when it diffuses into red blood cells, it will interact with the amino termini of the hemoglobin chains, and we have 4 hemoglobin chains, right? So each hemoglobin molecule would bind to 4 CO2 molecules. Now, forming a structure known as carbamate. Okay? So the formation of carbamate, again, you have a negative charge that is introduced, making it, again, increasing the probability of forming more electrostatic interactions, more or stronger T state of hemoglobin. Okay? So we have 2 mechanisms. One, affecting the pH directly, affecting the pH by producing protons, and number 2, by interacting directly with the hemoglobin molecule so that when it goes to lungs, it can be released and it goes out. So there are 2 mechanisms. So the question is, which one is the main factor? Which one is the predominant reason for the effect of CO2? And it turns out that actually 75% results from production of protons, 25% results from the formation of this carbamate. So there are 2 factors. Okay, this is actually, that's the plot in here. What they did here, notice, that's pH 7.4, no CO2. Now pH 7.2, no CO2, that's the effect of pH. Now right here, it's pH 7.2, so it's the same pH, but they increase CO2, and there is a shift. يبقى هذا right here, this shift from red to blue is the effect of the pH. From blue to purple, this is the effect of the CO2 directly, because the pH is the same. The only difference is CO2. واضح؟ واضح إن شاء الله? Okay, so there are 2 factors. هل أيوه اطلع. طيب. So what happens now in tissues is that there is production of CO2 from Krebs cycle. CO2 diffuses out from peripheral tissues. It goes into red blood cells. Some of it is converted to carbonic acid, and this carbonic acid, carbonic acid can dissociate into the ions, and it can diffuse out. So some of this CO2 is transported as diffuse, as a carbonic ion, okay? And some of it is linked or bound to hemoglobin, and that's how it gets transported into lungs, okay? So this is the effect of CO2. That's the second allostatic effect. Now there's something else, and there's a phenomenon known as isohydric shift. It means that there is an increase in the level of CO2, yet the pH stays the same. Blood is buffered. And the reason is that, well, hemoglobin acts as a buffer. Remember? It can bind to excess protons. It can bind to CO2, balancing or helping the bicarbonate system in buffering the blood. And this is known as the isohydric effect or isohydric shift, meaning that there is a change in the concentration of protons and concentration of CO2, yet pH is the same, iso. It's the same. Why? This is the phenomenon known as the isohydric shift. Right. So this is how CO2 is transported. 60% of CO2 is transported as bicarbonate ion. That's this. You have 30% of it is transported by binding to hemoglobin directly. And we have small percentage, about 10% CO2 as a gas in blood, because it's not really soluble in blood. Okay? So now, we talked about the effect of oxygen on affinity last lecture. We talked about Bohr effect, that is the effect of protons. Now we talked about the effect of CO2. So let's talk about the other allostatic effectors now. Continue with that. One effector is chloride ion. And this is a phenomenon known as chloride shift. So we have 2 types of phenomena. One, we have the isohydric shift, and the other one is the chloride shift. And the idea here is that, this is the wrong one, but what happens is that in tissues, when CO2 goes into the red blood cells and it gets converted to carbonic acid and it dissociates, some of the carbonic ion leaks out of the cell. That changes the total charge inside cells. So it has to be balanced. So as a result, chloride ions go in. So there is an exchange. Bicarbonate ion goes out of red blood cells, chloride ions go into the cells. So now there is no change in the total charge inside cells. This is known as the chloride shift. So how is that important? And the same, vice versa. The opposite is true in lungs. So what happens is when red blood cells reach lungs, CO2, or the bicarbonate, is converted to carbonic acid, carbonic anhydrase, converts it to CO2, and CO2 leaks out, and it's exhaled. As it goes out, chloride ions go into the cell. So here you have carbonic ion goes in, the chloride ion goes out, again balancing the total charge inside cells. This is known as the chloride shift. So what is so important about chloride getting into the cell? Well, once it gets into the cell, it can make interactions, electrostatic interactions, or charge interactions with the amino acids of hemoglobin, strengthening the release, or facilitating the release of oxygen from hemoglobin. Okay? So what happens again? Chloride, once it goes into the cell, like here. So as the chloride goes into the cell, there is reduced affinity between hemoglobin and oxygen is released into tissues. Okay? So when it goes into the lungs, chloride ions go out, meaning that hemoglobin becomes more in the R state. So it can bind to oxygen further. Yes, so that's the chloride shift. Questions? عيب مو تمام yeah. Okay. There is another major allosteric effect, and this is 2,3-bisphosphoglycerate. Now, 2,3-bisphosphoglycerate is a byproduct of the glycolytic pathway. If you remember, in glycolysis, we have production, glucose gets converted to glycerol phosphate, dihydroxyacetone phosphate. You have formation of 1,3-bisphosphoglycerate, and then you have formation of 3-phosphoglycerate. And it goes on all the way to pyruvate and then lactate, let's say. Okay. So, as a byproduct, we have the formation of 2,3-bisphosphoglycerate. So, instead of 1,3-bisphosphoglycerate, it gets isomerized into 2,3-bisphosphoglycerate. Now, there is a waste of producing 2,3-bisphosphoglycerate. The waste is that 2,3-bisphosphoglycerate can be converted to 3-phosphoglycerate, but there is no production of energy. When we convert 1,3-bisphosphoglycerate to 3-phosphoglycerate, there is production of ATP. When we convert 2,3-bisphosphoglycerate to 3-phosphoglycerate, there is no production of ATP. So there is a loss, but it is still advantageous. There is still an advantage of having 2,3-bisphosphoglycerate as a byproduct. ليش؟ لأنه what it does is that it binds to the core, to the center of hemoglobin molecule. When it does so, and the 2,3-bisphosphoglycerate has negative charges. If you look at the structure of 2,3-bisphosphoglycerate, it has a number of negative charges on the phosphate. So what happens is, when 2,3-bisphosphoglycerate sits in the middle of the hemoglobin molecule, it creates more electrostatic interactions because it has these negative charges. So it's almost like the way I imagine it. It's like you have a box, and this box is open. If you put your hand on it and wiggle it, does it wiggle? Definitely it does. It wiggles. It's not stable. But if you have a stick inside, it goes diagonal, goes like this. Can you wiggle it? You cannot. ليش؟ لأنه the stick would prevent wiggling the box. Can you imagine it؟ معي انتم؟ تخيلين الصورة؟ فاهمين علي؟ يلا يا جماعة interact, please interact. عيب. It's like a box. عندكم box. اوكي؟ بس مفتوح من جهة واحدة. هلا إذا حطيتي إيدك عليه، اوكي؟ وعملتي هيك، راح يتحرك البوكس؟ راح يتحرك. يس. لكن حطوا ستيك عصاية هيك، حطوها بالنص، diagonal هيك شكل. اوكي؟ من هذا الاند لهي الاند الثاني. Can you wiggle it? You cannot. ليش؟ لأنه العصاية ماسكة البوكس. Same, that's how I imagine it by the way. يعني ما قرأتها بأي كتاب ولا شيء, but that's how I imagine it. So the idea is that what this phosphoglycerate does, it fits into the core, the center of the hemoglobin molecule, and it makes it stiff, rigid. It's not relaxed. Okay, it's tight, it's tense, it's in the T state. So what this phosphoglycerate does is that it stabilizes hemoglobin in the T state, in the low affinity state. So is this phosphoglycerate important? Very important. Maybe it's one of the most important factors. Yeah, I'm not going to say the most, it's one of the most. ليش؟ لأنه this is hemoglobin with this phosphoglycerate in red, normal, sigmoidal. If there is no bisphosphoglycerate, look at it. It looks almost like myoglobin having this high affinity. So bisphosphoglycerate is one of those factors that stimulates the sigmoidal shape of the oxygen saturation curve. It's one of those factors that induce the formation of the R state and the T state. Okay? The allosteric shape of. طيب نعم. عادي صوتك. ليش ما؟ ليش بيكون أعلى البلاك؟ إنه عالي. بس لاحظ إنه بوصل على 100%. يعني هذه البوينت. هو الايديا انه انت بدك تروحي على ال 50% بغض النظر يعني وين، بس هم واصلين على نفس الليفل 100%. Okay? بس روح على 50%. To fill 50% of hemoglobin, you need less oxygen. So it's almost like حتى لو رحت على 50% وانزل تحت, it's almost like 5 torr, which is very close to myoglobin, really high affinity. Right? So what this phosphoglycerate does, it facilitates the formation of hemoglobin in the T state, facilitating the release of oxygen. And that happens in tissues. And it does make sense. In tissues, what do we have? We have metabolism. Glucose gets converted to pyruvate, right? During this pathway, the glycolytic pathway, as a byproduct, take that, we طالع على جنب ايش في عندنا؟ 2,3-bisphosphoglycerate. It leaks, it goes, actually it is in red blood cells, it binds to hemoglobin and it releases oxygen into tissues. Now, 2,3-bisphosphoglycerate is important when you go to high altitudes. So if you go to a mountain, so 5,000 meters, high, or if you look at people who live in high altitudes and you look at the level of 2,3-bisphosphoglycerate, they have higher level of 2,3-bisphosphoglycerate. ليش? To release more oxygen to tissues because there is less oxygen than at sea level. So if you go to a mountain, after 2 days, you adapt. Why do you adapt? Because your body converts or it produces more 2,3-bisphosphoglycerate. This is a way of adaptation. Okay? So that's the idea. That's how important it is. Something else, an important note about 2,3-bisphosphoglycerate is that during transfusion in blood banks, when they store blood in fridges for some time, the level of 2,3-bisphosphoglycerate goes down. It gets degraded. So when they give patients when they transfuse patients with this blood from the biobank, there is no 2,3-bisphosphoglycerate. So what they do is that they rejuvenate. That is, they have a special media. They have special solution that they add to transfuse to that blood from the blood bank to compensate for the loss of 2,3-bisphosphoglycerate. 2,3-bisphosphoglycerate goes down very quickly. Within 8 hours, it's gone. Okay? So they have to add a little more 2,3-bisphosphoglycerate before giving it to patients. So that shows you different metabolites. Same thing with ATP. There would be no ATP in blood. طيب عشان نخلص. هلا في عندنا the effect of temperature. It turns out that at higher temperature, there's a shift of the oxygen saturation curve to the right. Again, this means that there's lower affinity. And again, it's all logical. It makes sense. When it is hot, you want to produce more. When your body is heated, you produce more energy. Logically, it makes sense, right? Also, when you exercise, you want hemoglobin to have lower affinity so that it provides oxygen to tissues. You're exercising. So when you exercise, your body gets heated, and that increases the release of oxygen into tissues. So it's all logical. So now this is the effect of temperature. Two last points, two last slides. Fetal hemoglobin. Is there a difference between HbA and HbF? Definitely. Not only in structure, but in function as well. Okay? And it turns out that one of the differences between HbA and HbF is that there is a histidine in the beta globin that is replaced by a serine in the gamma globin. So histidine carries positive charge, and it's gone. You replace it with a serine. You reduce the number of electrostatic interactions that can be formed in this hemoglobin. As a result, fetal hemoglobin has higher affinity. And it does make sense. Again, it's all logical. It makes sense. Okay? Because you want the fetus to actually extract or take more oxygen than the mother. And that's why. So not only does the mother breathes fast and hard because of the weight, it's because the baby, the parasite, keeps on stealing the oxygen. Okay, that's why she goes, طيب, take the terror. Final point. Carbon monoxide. Carbon monoxide, we talked about it before, in a sense that it competes with oxygen, right, in binding to heme. And it has higher affinity towards oxygen towards heme than oxygen. And that's true. So if you look at hemoglobin plus oxygen versus hemoglobin plus CO, look at the P50. For CO2, it's much higher than oxygen. So yes, hemoglobin has higher affinity than oxygen. Okay? So is that it? Is that the only thing that CO does, that carbon monoxide does? No, it does something else. If you combine, that's hemoglobin plus oxygen versus hemoglobin plus oxygen plus CO, it turns out that, one, not only that if you add carbon monoxide, the level of saturation is reduced, the affinity of hemoglobin to oxygen increases. Yep, again, I'll repeat this. What carbon monoxide does is that it increases the affinity of hemoglobin towards oxygen. So it binds oxygen more tightly with higher affinity. But someone would say, that's a good thing. No, it is not good. Because when it goes to tissues, there's high affinity, it doesn't release this oxygen. So there are two factors that carbon monoxide does. One, it competes with oxygen in binding to heme. Two, it keeps hemoglobin in the R state. That is, it does not release oxygen, and this is the problem with pollution. This is trivia, hey, facts. If in inspired air, the air that we breathe, we have 1% CO in a room, okay, or in the environment, this is fatal. Just 1%, that's all we need, okay? Now the other thing is that due to pollutants, التلوث هذا عمالنا نشوفه احنا, usually the level of hemoglobin bound to carbon monoxide is about 1%. So in non-smokers, it's about 1% as a result of pollution. But in smokers, it's 10%. And if you look at, if you do a blood test, hemoglobin level in smokers, it's always higher than non-smokers. It's not a good thing, because most of this hemoglobin is not functional because of CO as well as all of the other toxicants that exist in cigarettes. Be smart, guys, be smart. والبنات من أرجيلهم وأرجيلهم. طيب, so if the level of, if 40% of hemoglobin is occupied by carbon monoxide, that's fatal. So ابني هيك حكى انه طلع مع شباب عنده بالمدرسه وواحد بده يتفصحن وزلمه فايش عمل؟ حط مش عارف كم سيجاره بتمه وقاعد يدخن. يورجيه زلمه طول القعده وهو حاطط راسه على الطاوله شاب مش مصحصح. يعني be intelligent, be smart. ربنا خلقنا عقل, don't smoke. طيب, that summarizes everything. يلا enjoy الهيموجلوبين.